And finally, we're going to talk about the different temperatures at which microorganisms grow. We find them growing over a large range. Some will grow at cold temperatures, some grow at warm temperatures, and then we're going to look at a couple of other situations in which we find microorganisms grow that might be kind of a surprise to you. So with temperature, um, the reason that organisms grow at certain temperatures is that is the optimal lo location, temperature-wise, for their metabolism and all of their enzymes that are going to work in those processes. So one of the terms that we use for some of these organisms is psychrophile. And these are organisms that grow as cold as 0 degrees Celsius up to 15. So we're talking about in the refrigeration temperature range. A freezer, water does freeze at 0. Most of our freezers are at minus 20 degrees Celsius. So we do have organisms that can grow in the cold, and that's where they're optimal temperature. If you get them at warmer, their enzymes are not going to work very well, if at all, and they're not going to be growing. In fact, it may even kill them. Another group of organisms is called mesophiles. These are the ones that are growing at 20 degrees to 40 degrees Celsius. To give you some perspective on that, we are, as humans, at 37 degrees Celsius. So if we're going to be infected by a pathogen, it's most likely going to be a mesophile because we're the optimal growth for them. You could also think, I put some pictures of some kittens here because their temperature is within that range. So you could think of cats as being mesophiles. Same thing with dogs, that sort of thing. Then we have another group of organisms that are called thermophiles. And here they're growing at rather warm temperatures, 45 degrees to 80 degrees Celsius. Remember, water boils at 100, so we're getting fairly warm. And you can see one of these colorful hot springs that we have in various locations on the Earth. And we do find microorganisms growing there, which we didn't expect to find. This graph here, or plot, kind of gives you a different perspective of these kinds of organisms. Here they have the psychrophiles. Down here below 0, you get a tiny bit of growth. And then as you increase the temperature, you get increased rate of growth until you get to where the optimum is on whatever organism is that you're looking at. But as soon as you pass that optimum, the rate of growth is going to drop because their enzymes don't work very well there. And then with the slight overlap, we have the mesophiles. Again, the same kind of curve, and then we get up here at body temperature and slightly warmer. That's going to be their optimum growth. One of the reasons we sometimes have fever when we have an infectious disease is your body's attempt to fight them off, and by raising your body temperature, you may actually get to the point where it slows down their growth. It's going to depend on the microorganism. And then we have thermophiles that are going at much higher temperatures. We're not going to find pathogens in there. We're not going to find pathogens in psychrophiles. Now, we do know some pathogens that while their optimum is in the body temperature range, they are still able to grow at refrigeration temperatures. And you may have heard of Listeria or Listeria monocytogenes. This is one of those organisms that does grow in refrigeration temperature. So you have to be concerned with ready-to-eat foods if they become contaminated. And it's a real headache for the food processing industry. Now, so that's it for the temperatures. I did skip over something that's called extreme thermophiles. Those are organisms that are growing at even hotter temperatures than that 80 degrees Celsius. Now here we have a discussion about pH. So most organisms are going to be growing in the 6 to 8 pH range, which we think of 7 as being our neutral range, and 6 to 8 is a little wider than that, but most organisms are going to be growing in that region there. Certainly the pathogens are going to be. But we do have some organisms that grow at low pHs, and here they're talking about below a pH of 5. And you think about it, beer is at 4.2, sauerkraut is at 3.5, wine at 3. These are all foods that have been produced by microorganisms. So the microorganisms have to be able to live in these lower pHs to make these foods. Now you can get down to a low pH low enough where they stop working, and that's the end of the fermentation process. Alkalinophiles grow up to a pH of 10, so we're looking here at the opposite end of the range. We're going to be more of a concern with acidophiles or organisms that are tolerant to acid, because those organisms are going to get through the low pH of our stomach and get into your intestinal tract and cause problems. Again, pathogens like our neutral pH typically. Here you see human blood's at 7.4, nice and optimum form. Yeasts and molds are some other examples of things that like acidic conditions. In fact, we can use that if we want to isolate yeast and molds away from bacteria. If, let's say we have a mixed population, we can use the acidic condition. So we take an agar plate and add acid to it and drop the pH. Now, another thing that we've come across is that microorganisms live in areas that have different salt concentrations, and some of them are referred to as halophiles. Halo refers to salt, philes means it likes or loves that kind of environment. And these are going to actually require the high salt concentration. You put them in a normal salt concentration, like an example would be the IVs that you're given in a hospital are at about 0.9% sodium chloride. If you put them in the halophiles in that, they're not going to do very well. They need much higher concentration. And we actually refer to these high salt concentrations as hypertonic. Hyper means a lot, and tonic refers to the salt concentration, so to speak. So it's a high salt concentration. And an organism that I'm giving you an example is called Halobacterium. They discovered it. I don't know if it was in the Great Salt Lake or if it was in the Dead Sea, but it was one of these areas where they have high salt concentration. And so they gave it the name salt bacterium, essentially. Now, there are organisms that we refer to as facultative halophiles, which means they don't require the high salt concentration, but they're able to live in that concentration of salt. And a perfect example of it is Staph aureus. You may have heard of MRSA, which is a form of Staphylococcus aureus, one that we don't like because it's resistant to a lot of antibiotics. It grows in a much higher salt concentration than we might expect. We think of an isotonic, meaning the salt concentration that's most closely matches what's in our bloodstream and what our cells like, is 0.9% sodium chloride. Staph can grow up to 7.5% sodium chloride, so much higher. And again, we can use that in the laboratory to help us isolate Staph aureus away from some other microorganisms.